The Cure Symposium poster. A scientific poster is a large document that can communicate your research at a scientific meeting and is composed of a short title, an introduction to your burning question, an overview of your trendy experimental approach, your amazing results, some insightful discussion of aforementioned results, a listing of previously published articles that are important to your research, and some brief acknowledgement of the tremendous assistance and financial support conned from others. If all text is kept to a minimum, a person should fully read your poster in under 10 minutes. The reasons researchers present a poster at a scientific meeting can include to highlight a new publication, to obtain feedback on an ongoing project, and to form collaborations with other researchers. Although you could communicate all of the above via a 15-minute talk at the same meeting, presenting a poster allows you to more personally interact with the people who are interested in your research and can reach people who might not be in your specific field of research. Posters are more efficient than a talk because they can be viewed even while you're not there. Many different components can be included on a poster. For the Cure Symposium, posters should include the following: an abstract, an introduction, a statement of hypothesis, the methods used, results, conclusions, future directions, acknowledgments, and references. Unlike a manuscript, posters can and should adopt a variety of layouts depending on the format of your graphs and photographs. Layout. Unlike a manuscript, posters can and should adopt a variety of layouts depending on the format of your graphs and photographs. As long as you maintain sufficient white space, keep column alignment logical, and provide clear cues to your readers about how they should travel through your poster elements, you can get creative. As an example, perhaps you might want to demote the unimportant sections that few people read to the undesirable real estate at the bottom portion of your poster, freeing up the right-hand column area for your stunning conclusions. Poster title. Titles vary significantly in the amount of detail they provide. A simple title is often effective. The approach and model organism can also be included in the title if done efficiently. The block for your title at the top of your poster should also include the official CU logo, which is available for download on Canvas. Do not perform a Google search for a logo. University of Colorado Boulder recently overhauled their branding, so you also must be careful not to use the MCDB logo shown here. Although it's visually very nice, it is not approved by the university for use by representatives of the university. The title should be centered, and author names should be included either in alphabetical order or in the order of contribution, so the first author listed contributed most significantly to the research. Abstract. When you are presenting your poster at a meeting, you'll probably be asked to submit an abstract. This abstract is for inclusion in the meeting catalog or abstract book. The Cure Symposium will also have an abstract book that includes abstracts for all posters presented. For the Cure Symposium, you will need to include the abstract on your poster. The abstract should briefly summarize your entire poster, a bit of background, a bit of methods, a summary of the results, and the significance of the work. The abstract should not be longer than 300 words. Perform a word count. It will be truncated if it is longer. Introduction. Get your viewer interested in the issue or question while using the absolute minimum of background information and definitions. Quickly place your issue in the context of published primary literature. Provide a description and justification of general experimental approach, and hint at why your study organism is ideal for such research. Unlike a manuscript, the introduction of a poster is a wonderful place to put a photograph or illustration that communicates some aspect of your research question. Use of images on posters helps the listener navigate the poster and pay attention to what you're saying, and helps you remember what to say. Begin your introduction with a strong statement of the problem and why your research is necessary. This is your chance to sell the importance of your project. Use of statistics is very useful here. Your audience will be composed of mostly scientists who are resistant to wild claims without numbers to support them. Second, introduce your audience to the compounds you have chosen. Why did you choose them? What does someone need to know to understand why your hypothesis is strong? Include a brief summary of important previously published information about your compound. Finally, conclude your introduction section with a brief statement about how you will be testing the compound and what you expect to find. Methods. Briefly describe experimental methods, but not with the detail used for a manuscript. Use figures and tables to illustrate experimental design if possible. Use flowcharts to summarize experimental steps or timing of experimental procedures. Include photographs or labeled drawings of the model organism. Mention statistical analyses that were used and how they allowed you to address the hypothesis. You should include the following detail: concentrations of drug or reagents used, strain of bacteria, time, for how long were your experiments run, temperatures, positive and negative controls, statistics used to determine whether the tested compounds are hits. Do not include an equipment used section. You can assume that anyone reading or listening to the presentation of your poster will know the basic equipment present in a molecular biology lab. Results. You should include data that accurately and efficiently summarize your observations: dose-response curves, positive controls, negative controls, timing experiments. Emphasize whether your experiment worked, including statistics used to define worked. The text should be supported by one or more figures that efficiently demonstrate your findings. Do not present multiple iterations of the same experiment. Average the results and report them on a single graph. The results section should not address your hypothesis or provide any interpretation of the data. Simply describe in text what you observed, referring to the presented figures. If you have no data, it is okay. You can present data that would demonstrate that your hypothesis is correct, emphasizing that the data are hypothetical. All images should be big enough to see detail at 6 feet away and at least 300 dpi. Crop images to emphasize what's important and don't leave too much white space. For graphs, remove horizontal lines and don't rely on default colors. You can change colors to create a theme for the poster. Do not use 3D graphs. If you use an image that's not yours, but is in the public domain, on the web without a copyright, you do not need to attribute it, but a website reference below the image is always good. The graph shown here has the following problems: horizontal lines are not removed, and too many numbers after the decimal point. 2 is usually sufficient. Do not interpret the results in the results section. Simply describe results as they are shown. Conclusions. Remind, without sounding like you're reminding, the reader of the hypothesis and result, and quickly state whether your hypothesis was supported by your data. If your hypothesis was not supported by the data, explain why. Discuss why your results are conclusive and interesting. Attempt to convince the audience of these points and the relevance of your findings to other published work and to real organisms in the real world. Alternatively, you may need to convince the audience that despite your hypothesis being incorrect, the data are still interesting and relevant. If you have no results yet, discuss the possibilities and what they might mean. Future directions. What would you do if you could stay in Discovery Lab for another semester? For another year? With unlimited funding and resources? Be clear about what you would like to do and what you think should be done by someone else. Be creative with this section and assume that you are not limited by the resources or funding in the Discovery Lab. References. Follow standard biology format exactly. Find journal articles that support the information you present, especially in the introduction section. Also, if you haven't read a journal article completely, for example, you could only find the abstract online, you may not cite it. You may have as few as 3 or as many as 10 references, depending on how much is known about the compounds you are testing. Acknowledgements. Thank individuals for specific contributions to the project: equipment donations, statistical advice, laboratory assistance, comments on earlier versions of the poster. Mention who has provided funding. Be sincere, but do not lapse too much into informality in this section. Here is some information that will be useful for this section. The principal investigator of our sponsor laboratory is Corey Detweiler, PhD. Our funding sources are the Howard Hughes Medical Institute and our department, the CU Boulder Department of Molecular, Cellular, and Developmental Biology. You should also acknowledge the chair of the MCDB department, Lee Neiswander, PhD.